In this video, we will show how to obtain selection signatures based on variation of linkage disequilibrium using the program varLD. If you don't know this program or how to do this entire process, don't worry, because I have you covered from the data through the program and through the entire script up until the visualization of the results. We will start with the presentation of the prerequisites. So a few things that you need to have in your computer before you start. Then we will go through the data modification step, because varLD requires a bit of a specific structure of the input files. Then we go through the analysis itself, and then at the end, the visualization of the results. As for the prerequisites, so I put them up here. So I kind of assume, as the viewer of the channel, that you have already PLINK and RStudio installed. Well, if not, then these things are also necessary. And also, of course, the tidyverse package. But on top of that, you need to have Java installed on your computer, just because the varLD is written in Java. Point number 2 here is to acquire the data. Of course, if you have your own data, that's fine, and you can go forward to analyze that. But if you don't have any data and you want to try out this process, then, well, this is the good time to download some data, for example, from the VIDA database that was presented also in this channel. So here is the link, but I will show it to you also a bit more detailed just in a minute. So the third point here is to acquire the program. I think there is a publication in Bioinformatics Journal, and there is a link, but that link does not work. But this link actually works, and it has everything what we need. Then there is a short manual, well, basically a README file that contains the instructions. We will look at that, and then we will follow up with the entire workflow as described in this script. So here is a short note on the VIDA database. So, well, I went for the cattle, and I chose the Charolais data, and I'm going to be contrasting it to the Holstein data. The reason I selected these two populations, because I want to contrast the beef versus the dairy side of the selection signatures. Well, I hope to find something in that direction. And of course, Charolais is one of the most well-known beef breeds, and the Holstein is the most known dairy breed in the world. In this analysis, I went for the 50K data, and I selected groups or data sets that are around the same size. So with 30 and I think 26 individuals each, so have it a bit of a balanced data set. This is the mentioned publication in the Bioinformatics Journal. So you see the program, the software is a bit older, from 2010, but still I like it, and I think it's a very neat representation what can be done with selection signatures in a very neat and easy way. Here is the mentioned link, but this link does not work, well, at least not for me. So I searched a little bit, and there is this other link where the program is located. So basically you go ahead and download it here. For the program download, it is packed, so you need to unpack, and also it mentions here that please refer to README program for instructions. Here is also an example data set if you want to try it out, but we will be not using this one, but we will be using our cattle data. And here below, there is also some files for the plotting. These scripts, namely this first one and the third one, are integrated into the big script as I will show you later on. And of course, the references that need to be cited for the publications. This is how our data looks like. So this is a pretty standard PET file, PLINK PET file for 26 Charolais cattle. And then this one is for 30 Holstein cattle. This is how the README file looks like. So this is basically the manual for the software. It mentions the necessary files. So you see there are these 3 jar files that need to be in the directory where you want to run it from. This synopsis is basically how you run the program. So you see it's fairly easy if you have the data prepared in an appropriate way. Now, this appropriate preparation of the data is mentioned in the description. And it basically says that needs 2 genotype input files with 2 different populational groups. The format of these files is mentioned here. and it should be one line for each SNP, where first column is the SNP ID, second column is the position, the third column is the genotype for animal 1, the fourth column genotype for animal 2, and so on. So basically here the orientation is flipped compared to the standard PET file where we have one line for each individual. Here they want one line for each SNP. So we need to deal with this in our data preparation step. Also here it is mentioned that the positions are assumed in ascending order, and very important, assumed to belong to one or the same chromosome or gene. Well, we are working on the chromosomal basis, but this also shows that the analysis with varLD needs to proceed for each chromosome separately. So you cannot run the entire genome in the single line or a single run. So we need to take care of this also in our script. And there are also specific requirements for the genotypes, how they should be coded. So the one homozygous should be coded as 1, the heterozygous as 2, the other homozygous as 3, and the missing one should be coded as 4. And again, here mentioned, each row represents one SNP, while columns represent individuals or samples. Later on, format of the output files, and then some options for the program. So first ones are for kind of basic, and the second half of these options are for targeted varLD analysis. Yeah, and this is everything that you have. So, well, we need to deal with this information and proceed further. It sounds complicated, but actually there is a fairly straightforward solution for all of these issues and problems. We are back in R, where we will start with the data preparation step. We start with clearing out the workspace and loading the tidyverse package. We also set the working directory, but you see that unlike the other scripts I'm using, normally I used here the path variable or set a variable name path to include the location of the files. This solution will come in handy later on when the same path will be used also for the visualization. Also, there is a variable for the number of autosomes because, well, we work with cattle right now, and it has 29 autosomes, but of course you can work or will probably work with other species which have different number of autosomes. And because varLD needs to be performed on a chromosomal basis, we need to perform these loops as many times as many chromosomes you want to analyze. And because this variable pops up in quite a few places, sometimes a bit hidden in the script, well, I decided to put it forward. So you need to change it just in this one place. So we run this first part along with the fairly standard quality control. So you see that we're removing SNPs and individuals that have a missingness more than 0.1 or more than 10%. We do a light control on the Hardy-Weinberg equilibrium, and that is basically it. And we are naming it breed1 underscore QC and breed2 underscore QC for the output files. The input files are, in this case, of course, the name Charolais and Holstein. Here, I would note that I try to make this script as general as possible, but of course, when you are implementing it for your own data, you need to make changes. And one of these changes is most likely the name of the input files will be probably different for you. Also, if you don't have PET and map files, you have binary PET files, then of course you need to change this to B file instead. Also, I will try to highlight all these necessary adaptations as we go, so I get you covered all the way. Now there is this next small section here, which kind of finds the SNPs appearing in both populations and then creates a common list of them that will be later extracted from the data. That's because the program contrasts exactly the same regions from the two populations, so that it makes sense that exactly the same SNPs appearing in both data sets. I think varLD does anyway something in this respect, but I am more comfortable to make it in advance, so I don't have to worry about it anymore. Now comes the data preparation process, and well, I will show it to you how it works on chromosome 1, and then we will run it for all the chromosomes. One thing I forgot to tell you before or show you before, just what are the files that are necessary to have in your working directory. So we have a bit more files here from the PLINK already, but it's still okay to show. So basically, these are the 3 files for the varLD. So there are 3, actually it's not 1 file, but 3 Java jar files. Well, this is the README we just read. The PLINK executable, well, also should be in the working directory if you use this script. And also then the data, for me, it's the Charolais ped and map file and the Holstein ped and map file. So these are these files that are highlighted right now. These are necessary to have in your working directory before you start this script that I'm just showing you. Okay, so we go forward with the script file. So we run this small part on the overlaps, which produces this common SNPs population 1 and 2, and proceed with the data preparation in chromosome 1. So just for the demonstration purposes, we take chromosome number 1 here. So I mentioned that varLD requires quite a specific structure for a single chromosome, but fortunately there is an option in PLINK that takes us quite close already to the final solution. And this option is the -- recode A transpose. We need to do this on a chromosomal basis. That's why we have the -- CHR and the dynamic chromosome number specified here. So we run this for PRED1 and chromosome 1. You see that there is a traw file produced, which we load promptly into R. And after opening it, we see that it's quite okay in terms of format. So there is chromosome, SNP name, centimorgan position, and then we have each individual here in a single column. There is a bit of a problem with the coding. So there is the one homozygous here is coded as 0, the heterozygous as 1, and the other homozygous as 2. And the missing ones, well, yeah, I needed to scroll a bit, but the missing ones are coded as NA, so missing or not available. So you remember from the manual or the README file that the AA should be called, or the one homozygous should be coded as 1, AB as 2, or the heterozygous as 2, and the other homozygous as 3. So we can solve this very easily if we add 1 to each of the numbers that we have seen before. And of course, because we want to add this number or this just 1 to the genotypes and not the other ones like chromosomes or position, we start from the column 7 onwards. So after this very quick step, we see that the format of the genotypes is already done, or well, in a correct way. So that is 1, 2, and 3 as required. We still need to solve the problem of the missing values, however, because the NA remains NA even if adding 1 to it. So what we need is to replace the missing or the NA values with the 4. We do this in this step and basically replacing NA with the number 4. Also, you might remember that only the SNP name and position is required. So we don't need all the other files, or rather said other columns here. So we just remove them and write out an output file that we will be taking along with the varLD program. So in this case, population 1, chromosome 1, varLD.txt. So this needs to be done for population 1 to prepare file for analysis for varLD. Exactly the same thing needs to be done with population 2. So basically extract the chromosome, recode with transpose A or A transpose, read in, add the number 1 to all the genotypes, replace the missing genotypes, and get rid all of the unwanted columns. And the output file population 2, chromosome 1, varLD.txt will be for the first chromosome. After running these parts, we will be ready to run varLD itself, which is itself a single line according to the manual Java jar, and its rgenetics1 jar is the actual program. And then what we want to have here is population 1 and population 2 with an output file name varLD chromosome 1.txt. You might notice that everywhere this is this dynamic chromosome number. That's because we want to perform all of this in the loop afterwards. There is one other thing you might need to do. I noticed that depending on your regional setting of the Windows, the varLD might produce file that has decimal commas instead of decimal points. Well, I know that this is customary in some countries, but of course, if this is created by default, then the R and well, other parts of the scripts will not work. So check the output, or if you have error messages, then check if this is indeed the case for you and change the decimal commas to decimal points, just uncommenting these 4 lines. And then you have these new output files for varLD, which is called .varLD chromosome number and .txt. Now, if you indeed need to use this, then just make sure that this file name is specified later on in the script on the appropriate places. I will highlight that place for you. But your default approach is to run the script, and if there is no problem or no error coming up, so you that just can forget about this part entirely. Now, this was already the part for the data preparation and running varLD itself for the chromosome 1. Of course, you don't need to do it for chromosome 2, 3, and 4, and so on, but you don't want to change these numbers manually all the time. So that's why everything is implemented in a neat little loop. Also, commenting out the loop and just running it for a single chromosome only might be a nice thing to do for test case. And when everything works, then you run everything at once. Here I showed you everything I wanted, so I just comment out this one, very important, and we comment in the loop. So this is the start of the loop. So the chromosome number will change from 1 to 2 to 3 to 4 and all the way up to the autosome numbers, which we specified up here. And so this is the start of the main loop, and then this is the end of the main loop that also needs to be on line 113 in this script. Needs to be also commented in. So between these curly brackets from line 113 and 48, so this will be executed 29 times, or as many times as many autosomes you have in your data. So I run the varLD for all the 29 autosomes. So you see the output file or the log file here. So there is 50 SNPs per window considered. Also, there is a bit of a math correction implemented directly. File 1 was population 1 for chromosome 29. Of course, this changed all the time. Population 2, chromosome 29. And the output file name is called like this. Also, a bit of a check on the number of individuals from 1 and 2 and the number of SNPs. The next step is actually standardizing the outputs or the LD values. And I took this part directly from the varLD website. So you remember I mentioned those 2 script files. Now this is their content, and I took their content and copied them directly here. So also this is the disclaimer. I did some changes, and the first change is just commenting out this line. So you remember also at the beginning of the script, I told you that I'm already implementing the working directory as variable path. That's because this variable path is also used in the descript from the varLD authors. And the other thing that needs updating perhaps is the file name, that is this varLD_chromosome. So this is how the output files are named. So you see here varLD_chromosome. So this is the base part of the file. These other parts will be added on by the script by the authors. Now, this is the part that if you used different file name, or you changed here the file name or after changing the decimal commas to decimal points. So if you have a different base file name here, you need to update it also here. But if you haven't done any changes to the output file name, so you can leave it as it is. And this last point here is the percentile of the genome-wide distribution to highlight. So these are specified by the authors, and I think they are quite good when we can keep them. So then this is the top 5% of the results, top 1% of the results, top 0.1% of the results, and top 0.01% of the results. This also needs to be done for each chromosome separately, but here we also have already the autosome number implemented. So this is what I told you, so that sometimes these things are a little bit hidden, so it is better just to change them at the beginning of the script, and then you don't have to worry about it anymore. So this part of the scripts is about the standardization as provided by the authors. And then after that, there is the plot for actually the plotting of the LD values. So this is also by the author. So this was again in a separate script, but I took it and pasted it here, including the disclaimer. Here I also changed some things or adapted some things. Again, there was a path here, which we already have. Now there is again the file name with the same name. So again, if you changed it before, then you need to adapt it here. Now I kept it here again, because you might want to run this part of the script separately at the end when you have everything ready, and because all the files are already on your hard disk. So you don't need to actually rerun the whole script all the time if you just want to visualize already existing results. So you can just run really just this part when everything is prepared and you just want to see some kind of results visualized or some of the chromosomes. So that's why I kept it here. Now the percentiles are the same. And here I did one more change, because before these two scripts, the standardization and this visualization, were separate. So we needed also varLD thresholds, and they needed to be supplied manually. But these varLD thresholds are actually computed by the standardization script before, and they are actually saved already in R. So to save you also a bit of a time, and also that you don't need to worry about this, then I take this varLD threshold straight away from the previous computation. So I have to amend a little bit my previous statement. So if you don't have this varLD threshold saved in R in some way in the workspace, then you actually need to rerun the standardization step and then the plotting of the varLD values if you want to just plot them and want to plot the existing results. But both of these scripts are really quick to perform, and well, you still don't need to rerun the entire varLD analysis all the time if you want just to plot existing results. Now, here is a bit of a sketchy part that I didn't know how to solve. So it starts, so it plots a region with a certain start and end base pair. And this could be really different for different organisms. Now, I didn't want it to interfere too much with this code or with the code of the authors for the plotting. So basically, I just specified here as starting position 0 and well, the region, and it's a really high value that should be okay for all the chromosomes. But if you are unsure if your organism has a really long chromosomes, you might want to look at this setting for the region and in particular. But other than that, the whole script is good to go. So first we run just the standardization, and you see, so these are these varLD thresholds I was talking about, computed for the 4 cutoffs or the 4 percentiles. And these are different all the time for different data sets. We run now the plotting of the LD values, and this part produces these types of plots. So basically, this is actually the cutoff, and only the top 5% is shown here. So this is for chromosome 29 and 28 and so on for each chromosome. So you see that at the actual thresholds, so this is top 1%, this would be top 0.1% are highlighted with these dashed lines. So you can have each chromosome separately. For example, this one would be a nicer or basically a higher peak. in terms of results. And because I ran this before, I know that chromosome 13 is the one with the highest peak. So you see here that so there is the highest selection signature on chromosome 13 for this particular data set and populations. And so this is the cutoff for top 1% of 0.1% and also top 0.01%. And this chromosome-wise plots are nice and all, but I thought it would be also nice to have a general overview what's going on. So that's why I also added in a plot of my own, or well, I just experimented a little bit, and we do a full Manhattan plot. And of course, Manhattan is written with 2 T's, which we correct right away. To do this Manhattan plot, we need to read in all the output files again and put them into one big data set in R, which we call full data. And then afterwards, we load everybody's favorite QQman package. So if you don't have this installed, you need to install this QQman for this particular visualization. And we will use the Manhattan function from this QQman package to do the actual visualization. Now, this Manhattan function usually is used for the visualization of GWAS results. So normally we visualize p-values, but after some convincing and after, well, just setting the appropriate options, most importantly, log p equals false, we could use it to visualize also the varLD results. And there will be 2 lines there. So it will be, the first one will be the varLD threshold 3. So this will be the 0.1% of the results. And the second threshold will be the 0.01% of the results. So this is the varLD threshold 4. So, and this is how the Manhattan plot looks like. So we see that there is a bunch of the chromosomes, but there is no high signals, at least when it comes to 0.1 or 0.01% of the results. But here are some of the chromosomes that deserve our attention. And of course, we can go on and see what are these regions exactly, where are they located, and what interesting genes are residing there. But for this video, we stop here and we just are happy to see some really neat results which came out from this varLD analysis. Let me know in the comments below how you like this video and perhaps what other software you want to see on the Genomics Bootcamp channel. But for now, I thank you for your time and have a very nice day.